I'm Jerry Ronan. I'm the CEO of Farfield Scientific. It's a company that was founded about 10 years ago, and we have developed a technique called dual polarization interferometry, which is used to study molecules and really how molecules change shape. And that's very, very relevant to life scientists.
I'm Professor Jeremy Turnbull from the School of Biological Sciences in the university. My lab works on a family of complex sugars called heparan sulfates, and a couple of years ago we discovered that these sugars actually are able to control an enzyme that turns out to be one that's very important, and this enzyme actually clips proteins into small pieces.
So it's implicated in things like various cancers. These functions are ubiquitous in a lot of biochemistry, and being able to study them and manipulate how they work is really quite an exciting field to be involved in.
The collaborations like the one with Farfield Scientific are of great benefit to us because it gives us a very close relationship with the development of new technologies. Instruments like those that they're developing are extremely useful to us for studying the interactions of sugars with proteins, and those kind of applications, understanding the fundamentals of the interactions of those molecules, leads to opportunities to develop new drugs for diseases like Alzheimer's, for inflammation, for cancer, other neurodegenerative disorders and so on. So it's actually very wide-ranging potential applications.
My name's Tim Lever. I'm the Chief Executive of Delphic Diagnostics. We're a specialist HIV diagnostics company based here in the UK. And what we're trying to do is take a suite of HIV diagnostics which will allow a clinician to individualise care out of the UK and across Europe.
Professor David Back, Professor of Pharmacology in the Biomedical Sciences School. My research group in the University of Liverpool started working in HIV in 1989. At that time there was only one drug for the treatment of HIV, a drug called AZT. Subsequently, new drugs emerged, so today we have 20 drugs that we can use for the treatment of patients. The focus of our team has been to try to optimize, to make sure that patients get the right drug for them. And one of the ways that we have done this is to develop methods for detecting the drug in the bloodstream of a patient, making sure that when a patient takes a drug, they have enough drug for their treatment, that they don't have too little or too much. And the process has been called therapeutic drug monitoring. So we monitor the amount of drug in a patient. And in 2003, we joined with Delphic in a licensing agreement whereby Delphic were promoting the therapeutic drug monitoring service for HIV patients, and we're going out to the hospitals to make sure that the system in place was a robust system for determining that. And then in 2005 we spun out the therapeutic drug monitoring scheme into Delphic as a company. And as researchers then we're involved in ensuring that the highest quality of new developments are also interfacing with what Delphic are doing and the plans for Delphic, so that we really do make sure that the research is impinging on where we can make an impact on the individualised care.